A sudden, involuntary pause in the middle of a sentence is not merely a social awkwardness, it is the observable output of a fast, hierarchical decision process in the brain that temporarily suppresses motor output to protect goals that are more costly to gamble with than to delay. At the moment of freezing in conversation, multiple systems converge, threat detection computes the potential cost of speaking, cognitive control monitors conflict and error likelihood, motor planning circuits are inhibited, and autonomic pathways adjust breathing and heart rate. The freeze is a defensive computation, an implicit risk assessment that favors withholding action while uncertain information is resolved. The mechanism begins with rapid evaluation of social context. Sensory cortices feed facial expressions, vocal tone, and semantic content into networks that estimate social threat, whether the current turn-taking, the interlocutor's expression, or a remembered social rule raises the probability that a spoken response will cause harm to reputation, status, or relationships. The amygdala and connected limbic structures act as early detectors of salience and negative valence. They signal to brainstem areas and to the anterior insula about unexpected, potentially costly stimuli. Parallel to that, prediction error systems, centered on the anterior cingulate cortex, ACC, register discrepancy between expected conversational outcomes and incoming evidence. When prediction error grows and expected reward of speaking diminishes, the ACC signals the need for heightened cognitive control. Cognitive control is implemented primarily through prefrontal networks, especially dorsolateral prefrontal cortex, dlPFC, and ventrolateral prefrontal cortex, vlPFC, which mediate working memory, selection among competing responses, and inhibition. These frontal regions can suppress motor plans by influencing basal ganglia output in supplementary motor areas, which are responsible for sequencing and initiating speech. The basal ganglia operate as a gating mechanism. When the direct pathway facilitates action, speech proceeds. When the indirect pathway or hyperdirect pathway, via the subthalamic nucleus, becomes predominant, initiation is inhibited. The hyperdirect pathway, recruited by rapid conflict detection from medial frontal areas, can produce fast, transient pauses that align with the phenomenology of freezing. Autonomic modulation accompanies and enforces this motor inhibition. Parasympathetic output via the vagus nerve often produces heart rate deceleration and changes in breathing that make phonation more difficult. Concurrently, the locus coeruleus norepinephrine system increases arousal and narrows attentional focus. The polyvagal framework links these processes to social engagement. When the ventral vagal complex downregulates, muscles needed for expressive speech and facial engagement lose their tonic support, making vocalization harder and reinforcing silence. Neurochemically, surges in noradrenaline enhance vigilance but can overload prefrontal circuitry, while acute cortisol elevations impair working memory and lexical retrieval, both of which increase the probability that an attempted utterance will stall. At the level of language production, freezing often results from failures in lexical access. Broca's area and adjacent left inferior frontal regions coordinate retrieval of semantic and phonological representations stored in posterior temporal cortex. Under stress or high uncertainty, these interregional interactions become less efficient. Weakened recurrent activity in temporal language areas reduces the signal available for frontal selection, and concurrent inhibitory input from medial frontal conflict signals suppresses the motor program before phonological encoding is complete. The subjective experience of losing the word or of a gap between thought and speech therefore emerges from a cascade: degraded retrieval, frontal detection of increased error risk, and motor inhibition enacted to prevent an inaccurate or socially costly utterance. This cascade embodies a cost-benefit computation. Freezing trades immediate communicative fluency for reduced probability of a damaging utterance. The cost side includes reputational damage, social rejection, or escalation of conflict. The benefit of speaking may be small under high uncertainty. Evolutionarily, defensive immobility reduces exposure to immediate threat. In social mammals, withholding vocalizations that could trigger aggression can be adaptive. The brain implements a time-limited withholding strategy that buys milliseconds to seconds for integrating additional cues, a second glance, a clarifying expression, or retrieval of a safer turn. If new evidence reduces uncertainty, frontal systems re-enable motor plans and speech resumes. If not, silence may persist or other nonverbal behaviors may be adopted. Despite this coherent picture, several important constraints and weaknesses limit current understanding. First, the mapping from animal models of defensive freezing to human conversational freezes is imperfect. Most basic research manipulates predator threat and measures immobility. Human conversation involves complex, abstract social valuations that are difficult to model in rodents. Neural substrates are partially conserved amygdala, PAG, ACC, and descending autonomic pathways, but the human frontal language network adds unique layers that complicate causal inference. Second, much evidence comes from correlational methods, fMRI activation patterns and heart rate changes associated with public speaking where social evaluation tasks illuminate candidate regions but do not prove causality. Lesion studies and transient disruption with transcranial magnetic stimulation provide stronger evidence but are sparse for the precise circuits involved in the instantaneous speech interruption. The basal ganglia and frontal subcortical loops implicated in motor inhibition are deep and difficult to perturb non-invasively, leaving mechanistic gaps. Third, physiological trade-offs create edge cases where the freezing response is maladaptive. In urgent, high-stakes interactions, medical triage, military communication, or rapid team coordination, freezing can produce catastrophic delays. Conversely, in chronic conditions like social anxiety disorder or major depressive disorder, the freeze mechanism can become overactive or improperly gated, leading to persistent avoidance and impaired social functioning. Pharmacological modulation illustrates the fragility of the system. Benzodiazepines reduce anxiety and may lower freeze propensity by dampening amygdala output but also impair working memory and blunt appropriate social signaling. Beta blockers reduce peripheral arousal without addressing cognitive evaluation, which may leave the underlying cautious decision process intact. Individual differences further complicate predictions. Baseline working memory capacity, trait anxiety, cultural norms about silence, social status, and prior learning about social contingencies all alter threshold settings for suppression. Developmental factors matter. Children and adolescents have immature prefrontal control and different social valuation metrics, so freeze responses have different frequencies and consequences across age. Neuromodulators such as oxytocin interact with social salience networks in context-dependent ways. In some contexts, oxytocin increases social approach and reduces freezing, while in others it heightens sensitivity to negative social cues and thus increases inhibition. Measurement limitations constrain scientific progress. Naturalistic conversation is dynamic, multimodal, and context-rich. Laboratory tasks that simulate social threat often sacrifice ecological validity for experimental control. Real-time neural and autonomic recordings during spontaneous dialogue are technically challenging. Motion artifacts confound EEG and FNIRS. fMRI is incompatible with free speech, and invasive recordings are limited to clinical populations. That leaves a gap between controlled experimental findings and the lived experience of freezing in real-world interactions. Looking forward, a central unresolved scientific and engineering challenge is to move from descriptive correlation to causal, predictive models that operate in naturalistic settings and can distinguish adaptive transient freezing from maladaptive chronic inhibition. This requires integrating 4 strands. First, development of minimally invasive, portable neurophysiological measurement systems capable of capturing rapid interactions among frontal control signals, limbic salience markers, and autonomic readouts during real conversation. Advances in wearable EEG, scalable FNIRS, and high-fidelity peripheral sensors may close this gap, but signal processing methods must handle speech-related artifacts and multimodal alignment. Second, mechanistic computational models that unite predictive coding accounts of social inference with control-theoretic descriptions of motor gating are needed. These models must represent uncertainty explicitly, compute expected costs of speaking versus withholding, and implement a gating variable that reflects basal ganglia thalamocortical dynamics. Such models would generate testable predictions about when and why motor plans are suppressed, linking observed pauses to latent variables like prediction error, decision threshold, and working memory load. Third, causal perturbation studies using neuromodulation, targeted pharmacology, or precisely timed transcranial methods must validate the model's components. Ethical constraints limit interventions in healthy individuals, so carefully designed studies in clinical populations or combine non-invasive brain stimulation and computational modeling will be required to establish which nodes are necessary and sufficient for freezing in conversation. Fourth, translation into interventions demands distinguishing context-appropriate freezing from pathological inhibition. Behavioral training that shifts threshold settings, real-time biofeedback that signals when autonomic patterns predict a freeze, and closed-loop neuromodulation that augments prefrontal control transiently could help selectively reduce maladaptive pauses without removing adaptive caution. Each approach raises safety and ethical questions. Altering social risk assessment has consequences for consent, vulnerability to misinformation, and cultural norms of deliberation. The momentary silence that can paralyze a conversation reveals a protective architecture: rapid valuation, conflict monitoring, motor gating, and autonomic enforcement. Understanding how these layers interact in milliseconds is both a scientific frontier and an engineering problem. The unresolved question is whether it is possible to build predictive, causal systems that monitor natural conversation in real time, accurately infer when a freeze reflects adaptive caution rather than dysfunction, and intervene in a way that preserves social prudence while restoring fluent communication.